Hi, I'm Jamie Costello, founder and CEO of Genomics. In this video, I'm going to tell you a bit about genomics and epigenetics, which are the scientific foundation for our individualized programs for nutrition and exercise. First, let's talk about genes. Genes, as we think of their involvement in inheritance, were first discovered in 1869. Though at that time they weren't called genes. People believed that genes were fixed and could not be changed. And while this is true in a strict sense, we now know that genes can be turned on or off based upon other factors. We now know what those factors are, and scientists are studying them. Since the entire human genome was mapped in 2001, the research in genetic science has exploded and offers many opportunities to improve health outcomes and to detect, understand, and cure disease. What do we know now? First of all, we needed to determine how a single fertilized egg can make hundreds of different cell types. Each cell has the same genetic code, and yet there are so many different functional outcomes. For example, liver, skin, eyes, etc. What we've come to know is that there's information on top of the genes that allow the formation of each functional cell type from that set of instructions. These instructions are called epigenetic marks, from the Greek word epi, which means upon or on top of. The human genome has 25,000 genes in each cell, but not all of these are turned on in each cell type, like in our example, the liver, skin, and eyes. Each cell has its own function, and the genes required for that function are turned on, and the others are turned off. A good example of epigenetic marks is with identical twins. If you have genetically identical twins, that is from a single fertilized egg that divides into two, despite being genetically identical, the twins will have slightly different appearances. Their height can be different, and they can have many other traits that would be different, although not always readily apparent. They both can carry a disease gene, but maybe only one gets the disease. In fact, the likelihood that they both get the disease is only about 20 to 30%. These changes in genetically identical twins result from these epigenetic changes that turn genes on and off. These epigenetic differences increase over time and are reflective of different exposures, such as diet and the environment. Today, epigenetics is not only used to describe the difference between identical twins, but more broadly, many nutritional and environmental influences on gene expression. There have been several large studies showing the effects of epigenetics. In fact, scientists have now made the correlation between disease as being brought about by epigenetic changes, including obesity, diabetes, cardiovascular disease, Alzheimer's, autism, and cancer. We know that epigenetic factors can be modified, and the FDA recently has approved 4 new drugs targeting these epigenetic factors to treat cancer. These drugs have been developed to turn on the tumor-suppressing genes that cancer turns off and to turn off the growth genes that cancer turns on. In addition, these epigenetic marks are also being used as biomarkers for early detection of cancer. Well, what does all this mean to you? All of this research has made it easy and inexpensive for you to get your genetic profile from companies like 23andMe. All that's required is a simple saliva test you can do at home. From your raw genetic data, my company, Genomics, can analyze 260 specific gene variants and prepare a detailed report for you with the best diet, supplements, and exercise program for your body and its metabolic factors. I urge you to call me at the number on our website or email me at the contact page to find out how easy it is to get started. Truly, there is no better way to achieve your health goals than this.